Thanks so much, Patrick, and thank you all for joining. This is the final webinar in a three-part series dedicated to effective partnerships with external research or manufacturing organizations. So today our focus is on chemistry, manufacturing, and controls. And we're very lucky to have one of my colleagues, Dr. Mark Levy, presenting today's webinar. Mark is an expert in regulatory affairs with over 20 years of experience. He will strive today to answer common questions about required CMC activities by providing real-world examples, as well as really talking you through the benefits of external partnerships. So with that, Mark, you are free to start.
Okay, thanks, Patrick and Marissa. Good afternoon and good morning. As was discussed, this seminar is on the considerations for external partnerships, CMC. So let's look at first what the objectives are. The presentation will aim to address the use of contract organizations in the development of drugs and biologics. These will include CROs, which are contract research organizations, CMOs, contract manufacturing organizations, CDMOs, which are contract development and manufacturing organizations. Now, there is no one-size-fits-all approach to outsourcing drug development, and cost estimates vary greatly from organization to organization and are also highly dependent on the complexity of the activity and the type of drug or biologic. In this talk, I have combined, where applicable, drugs and biologics. Sometimes they will be referred to as drugs and biologics, sometimes one or the other, and sometimes therapeutics. So let's look first, and this number always tends to grow, at the estimate of the investment needed to bring a new therapeutic to the US market. The CBO, which is Congressional Budget Office, a nonpartisan group who costs projects, legislation, etc., came up and said that the estimated cost of developing a therapeutic and going to market ranges from under $1 billion for some biologics to well over $2 billion for some drugs. I will date myself and say I remember that when that number was 750 million, and that was for drugs. Now, small drug companies, which are those with less than 500 million in revenue, account for greater than 70% of the 3,000 drugs in phase 3 trials currently. And about a third of the new drugs approved have been developed by pharmaceutical firms with annual revenues of under 100 million since 2009. Now, large drug companies, those over $1 billion in revenue, still account for greater than 50% of the drug approvals since 2009. Meanwhile, the likelihood of an approval from a phase 1 trial remains still at roughly 10%. So 1 in 10 that go to phase 1 wind up approved. Excuse me. So let's look now at the purpose of CMC, which is the shorter version of chemistry, manufacturing, and controls. First, the drug or biologic must be manufactured. You also need to assure that the drug or biologic sold to the public will have quality attributes similar to those of a drug or biologic demonstrated to be safe and effective. That is the one that went through the clinical trials. Also, you must assure that the quality of the drug or biologic meets appropriate standards and is consistent and that is consistent quality and a consistent product. And you need to be able to assure that the drug or biologic you are using is the drug described on the label. And recently there was a case where a drug entered phase 3 and it was not the structure that had originally been presented to the FDA. This comes into play when you have multiple chiral centers for a small molecule, and biologics are a different ballgame altogether. So CMC is governed in law. The legal basis is 21 CFR 312.23A7, where the CMC concerns the composition, manufacture, and control of the drug substance and the drug product. Now, the two terms that are coming up, drug substance and drug product, each have a definition, which I will go through, and the legal citation for the definition is noted in the slides. Biologics still have a drug substance and a drug product. You define them. If you're developing biologics or vaccines, you define the drug substance and the drug product, but they still have a drug substance and drug product, even though they're biologics. Drugs obviously have one as well. Now, what is the drug substance according to the legal definition? That's the active ingredient. It furnishes the pharmacological activity or other direct effect in the diagnosis, cure, mitigation, treatment, or prevention of disease, or to affect the structure or any function of the human body. It does not include intermediates used in the synthesis of such ingredient. This is commonly called the API. In biologics, this has different definitions. The drug product means the finished dosage form, tablet, capsule, solution. It contains an active drug ingredient. It does not have to have inactive ingredients in it, but generally it does for stability, to preserve sterility, lyophilization, salts, buffers, time release mechanisms, you name it, it's probably in there. Bulking agents, etc. The term also includes a finished dosage form that does not contain an active ingredient, but is intended to be used as a placebo. And this is the vehicle. You can think of this as a vehicle control for the placebo. Excuse me. In the biologics and vaccines world, the drug substance will be the serotypes that are added to the vaccine. An example is Prevnar 20 pneumococcal for 20, it has 20 serotypes in it. Each one of those has a drug substance. The drug product is the final vaccine that's injected into the patient. So now, in terms of certain cell therapies, there may not be a drug substance, because cell therapies, once they are produced, the solution they are in has to be intravenous grade, human grade sterile. They cannot be sterile filtered generally. And so you merely have a drug product. And everything goes to the drug product. So this is one time where there may not be a drug substance, but that's only in the biologics world. And FDA has no issue with that. You just explain to them why, and they're like, okay, fine. So if we look at planning for success with the right expertise on the CMC team, what do we need? We need generally CMC consultants, or a small number of employees with expertise covering several disciplines. Most of these shown in the wheel occur at the same time. The chemical development, the further analytical development, pharmaceutical development generally occur almost simultaneously. Solid state characterization has to occur, followed by the synthesis of the drug substance, which will produce the toxicology supply, the clinical supply, and the regulatory dossier. With the regulatory dossier being just as important as the chemical development, because that is what the regulators will review, be it the FDA, EMA, or whichever regulator you submit it to. So what are the critical elements of CMC? Well, how and where is the drug or biologic made? How are the raw materials tested and monitored? What control procedures are in place to assure product consistency and quality? Are critical quality attributes adequately identified and characterized for the product? You'll frequently hear these as CQA. Are the test methods used to monitor product quality appropriate? And what controls or how long does the product maintain its quality after its shelf life? This is stability, and that allows you to put the expiration date on the batch of material. So let's look at one of the key considerations, which is control of raw materials. As a drug developer, when you start going to clinical trials, you become responsible for the drug that is the subject of an IND application. So not only for the chemists here, do you have to consistently manufacture the drug substance, and then formulate it to drug product, you have to do this with pharmaceutical grade materials. The need for USP material can generally be met by the suppliers. Now, this is merely as an example. Fisher, or now Thermo Fisher, and Sigma Aldrich are two. They are not the only manufacturers, but they do sell USP grade material, starting materials. When you begin drug substance manufacturing, everything has to be USP grade, or you have to qualify the material if you can't obtain USP grade. Now, what if it says USP and EP? That's European Pharmacopoeia. Okay, fine. What if it says EP only? And that's the best you can get. You will need to look at a USP spec sheet and make sure that the material you are using exceeds the USP specifications, and explain that to the FDA in the IND filing. This happens frequently, and this has to do with the way the US law is crafted, not that the EP material isn't good enough. Frequently, EP standards are higher than USP standards, but the way the law is crafted, the USP is the only one whose standards are accepted. You just will list out that the material is EP grade, and then explain how it meets or exceeds USP standards. Now, what is the difference? Because if you go into the catalogs, you will find ultra grade material that may be a higher percentage of purity than USP grade. Well, in short terms, the excipients aren't harmful. The USP grade material may be 96%, but the excipients are known, they're permitted, and they're not harmful. At 99% of somebody's ultra grade, which will be great for a bioassay, the 1% that could be an impurity could be toxic. It'll work in the bioassay, but when you give it to humans, it could be toxic. So that is why. Also, you want to avoid materials of animal origin and or obtain a BSE, TSE certification and perform a use test because you're responsible. You also will want to identify and qualify multiple suppliers of key raw materials. And that is because there can always be a shortage, as we just saw during the pandemic. Suppliers can go out of business, or some company who needs 50 kilograms goes in front of you when you only need 500 grams. If there's a large order, you might get put to the back of the list. It can happen. Now, another key consideration is analytical testing. You will need to define the critical quality attributes of the drug substance and the drug product. What part is critical? These are characteristics that should be maintained within a limit or tight range to ensure desired product quality. The analytical methods you choose must be able to identify and confirm strength and purity of the drug substance and the drug product manufactured. And analytical testing can involve both in-process testing and release testing of drug substance and drug product after manufacturing. So you will hear someone talk about the in-process controls. And those are used as the process is ongoing to ensure that the quality is present and that the impurity levels aren't running too high. Additionally, CMC testing is product specific. For sterile injectable products, you will have to, in addition to all other tests, test for sterility and endotoxin concentration. For a controlled release product, the release profile of the active ingredient over time. An oral tablet, the dissolution profile. A soluble powder for drinking water, moisture content and solubility. An inhaled product will require testing for particle size. A cell and gene therapy product, I have here sterility, also dead cells and empty capsids. A transdermal product is the release profile. And though it's not listed, a lyophilized powder is also a dissolution time and particle size. Now, this slide goes into phase appropriateness of the cGMPs. And what we have is a case where what applies when? If we start out at the R&D phase, apply good documentation practice, GDP. Frequently you hear all the good X practice as abbreviated as G small xp. And GXP is anything that applies, be it good clinical practice, good tissue practice, good laboratory, good manufacturing, and good documentation. So at the R&D phase is good documentation practice. At the preclinical, where you're starting to do toxicology study, you apply some GLPs. Now, that means there can be non-GLP tox studies, followed by the GLP tox study, which is right before the IND application. You start the validation life cycle approach closer toward phase 1. Small molecules at phase 1, they're GMP products. Biologics get an exception provided it is close to GMP. Because sometimes with biologics, you cannot get the material at phase 1 to be GMP. You just don't. You would either have to spend 3 more years, or you would have to qualify all the starting material. So there's an exception made with Center for Biologics. But small molecules, GMP starts at phase 1. Then you start with analytical method validations on calibrated and qualified and validated equipment, increasing GMP expectations. And then toward phase 3, you start hearing about validating the manufacturing line. This is when, and I don't want to get into this too deep, you start hearing the words 3 PPQ batches, process performance qualification batches. And this is the validation of the manufacturing runs. Of course, the stringency of the cGMP compliance is expected to evolve over the course of clinical development. General cGMP principles applicable to phase 1 are sterility assurance, quality oversight and facility control, and adequate documentation. With ongoing clinical studies, you get into process and method validation and process controls within process testing. And then you start setting specifications. These come later. But if you produce 3 batches early on before your non-clinical studies start, keep whatever data you have available and ready so that when you start to write this up for the FDA, you can show where all the batches went and that your quality is either being maintained or increasing and where the batch went. We used it to test, we used it in pharmacology, batch disposition. FDA loves to read a batch disposition table. Should there be a difference between 2 batches, they want to know. You want to know, and they'll want to know. So, what are some more key considerations in manufacturing? Well, define manufacturing processes that are readily scalable. Consider changes in equipment processing parameters between laboratory, pilot, and commercial manufacturing scale. And define the critical process parameters and determine proven acceptable ranges. The take-home message from this slide is you will have additional requirements and critical process parameters as you go up toward larger and larger batches, be it a pilot scale or commercial manufacturing. You will also probably have to change the synthetic process, given that ones used in laboratories on a smaller scale don't always scale up perfectly. And as those of us who are chemists know, too often the reagents used in the laboratory do not go over well at the FDA. They're toxic they're explosive, and they work in a lab. But when you get to doing this on a large scale and having to purify the material and remove all last traces of impurity, aka reagents, you can't have toxic reagents sitting around. Now, there have been whole week-long seminars on process validation. Just know that eventually you do wind up having to go to validation. And that is where you prove that a process is capable of consistently delivering a quality product or that an assay is repeatable. You will also hear mentioned about QA, quality assurance. Among their other activities and oversight, they release the reagents, the starting materials, the drug substance, and the drug product for both non-clinical and clinical use, and they sign the final COA. They have other roles, and there have been week-long seminars on QA as well that we just don't have time to get into. Only to say that QA's advice will be to retain samples of non-clinical and clinical batches for standards for comparison as the development progresses, and store these samples under very controlled conditions so that you can go back and prove that what you made is exactly the same that you did your pharmacology testing on a year ago. Basically, it's considered your reference standard. Now, let's look at these CMC contract organizations. What is a typical contract manufacturer's services? They manufacture stable intermediates, the drug substance. They produce a drug product based on the batch formula. You may not always know what they do in the drug product world because frequently pill formulations and time release mechanisms can be proprietary, and you merely pay a fee for service. The product comes back with a guarantee that it dissolves over time as it's supposed to, and you get the rights to cross-reference a drug master file at the FDA. This is frequently done beyond phase 1, but you'll see it occasionally. And as you get toward phase 3, you'll see it more often. Contract fill finish, final mixing, filtration, pH adjustment, vial and bottle filling. There are contract packagers who also label vials, cartons, containers, package, blister packs, especially for clinical trials where the placebo needs to look exactly like the pill with the investigational drug in it. Otherwise, it's pointless to blind the trial if everybody can figure out which tablet is the drug and which tablet's a placebo. They conduct lot release testing on stable intermediates, drug substance, drug product, final bulks, which are used in biologics manufacturing, and our overall final product. And they do stability and sterility testing. Excuse me. Typical CDMO services, contract development and manufacturing organization, they tend to be more flexible than routine manufacturing, which allows a designer to fabricate one component of the product or take a concept and turn it into reality. They are justified as more economical and efficient to outsource than to invest in people with high competence, equipment, and resources. Do you want to hire it done, or you want to have to build your own pharmaceutical company? And a CDMO handles the outsourced manufacturing of drugs and biologics, as well as all of the innovation and development work that occurs prior to GMP manufacturing. And a lot of this has to do with formulation. So what are the vendor selection criteria? The big four are expertise, reliability, flexibility, and knowledge and information. The reliability, proficiency at implementation, do they do what they say they will do when they say they will do it? Flexibility, can they modify plans in line with project requirements? Knowledge and information, are they clear on the scope? Do you get updates on progress? Do you get recommendations? And do you get reports on development and manufacturing work? And for expertise, the technical, the regulatory, and are they innovative? How do you choose? Well, you look at expertise and experience. Is there a specific area you need support in? Is it conducting the trial or is it running the assays? And you assess their track record or the types of clients they've worked with in the past. They will tell you sometimes who their clients are or have been, what they've done, who they work with, the fact that they hold 5 drug master files, their quality and compliance. Do they have a strong quality and compliance department? Do they show that they have the compliance? You review their quality management system, their SOPs, and any certifications they have. They should be competitive and provide real value for money because they do this for multiple companies. And if they're too out of line, you'll go to their competitor. And you ask for cost estimates and compare them to other CROs. Now, do not be surprised if they have a minimum batch size. They may not be willing to produce 500 milligrams. It may be a 5-gram minimum. So don't be surprised at that. But if one will produce 5 grams and one will produce 50 grams, it may be more cost effective to get the 50-gram, provided you get it on stability, so that you can rest assured that you have enough material to get through the GLP tox study. Are they communicative? Do they collaborate well? Is there timeliness and flexibility? If your clinical trial is delayed, can they hold up? Can they start up if you're running early? What is their availability? What does their docket look like? Or can they meet deadlines? I have seen clinical trials be delayed for 6 months or a year because the CMO, who shall remain nameless but was one of the finest in the world, just could not produce material fast enough. And so the clinical trial had to sit on delay, but it was worth it to have that CMO produce the product. So it does happen. Where are they located? If you are developing a product for multinational trials, you will need to make sure that the starting materials are USP grade and EP grade as well, so that the material will suffice for both countries, European jurisdiction, regulations, because you need to satisfy US regs and European regs. This is where the ICH guidelines or guidance comes in very helpful, in that frequently you can use the same material for both. It's extra paperwork, but it's doable. You need reagents qualified, import-export issues. Sometimes it is difficult to export or import EP material into the US. It's you at that point you have to talk to your manufacturers about how they do it. Sometimes USP grade material being shipped into Europe can be an issue. But this gets into very specific questions that are beyond this seminar. Comparing global CROs and local CROs and assessing how much supervision will be needed can help you understand whether location will be important. It may not be, and it may be. So what are the GMP requirements for vendor qualification? Well, your selected vendors are evaluated for compliance against the regs. The results of the audit reviewed and need for a site follow-up visit. Site qualifications are done on a cyclical basis, generally every 2 years. And the stringency of process increases as the development program advances. So what are the benefits? The ability to evaluate systems vendor uses to produce a regulated product, and you can identify gaps in the quality system as it's ongoing and ask for corrective action. This is the CMO audit process, which I will only talk about lightly. At stage 1, there's an audit questionnaire of a potential CMO. Are they critical or not? Are they a service provider or a core pharma manufacturer? And is it qualitative risk or quantitative risk? If it's high risk, you do a remote desktop audit and you audit the CMO and you review until a residual risk is down to an acceptable level. And your consultants can help you with this. If it is very high risk, you could conduct an on-site audit. And so you just keep working through this until you get the risk level down to what's acceptable. And acceptable, you have to define acceptable, and you talk to your consultants about it. You keep going. Now, in addition to this, should your CMO get a warning letter, that puts you in a dire predicament because you cannot file an NDA with the CMO under a warning letter. So you wind up having to use technology transfer and get another CMO to produce the product. This is why you need a lot of documentation just in case it happens. So what are the types of agreements with a CMO? There are private label agreements, which are long-term contracts for producing part or all of a product, where the CMO creates the products that the business sells as their own with their brand name. Private label manufacturing, end-to-end manufacturing, and select component creation. You have agreements for services and materials. They involve deals for labor and access to equipment. The CMO's team and facilities used, but you run the manufacturing operations. Not all CMOs use the same equipment materials, and agreements may be needed for more than one CMO to acquire the best quality for different things from each party. And there are international agreements. Your third-party suppliers overseas. You do run into some unique issues for packaging and shipping into the United States, and I want to add also into Europe. And it may prove less expensive depending on your specific requirements. So what are the benefits of a CMO? Cost savings, time savings, access to advanced technical skills, GMP qualified facilities, a track record of regulatory inspections, internal product knowledge. But you as the developer need to ensure that the signed master services agreement covers all your intellectual property, quality, time frame, and other needs. Generally, you get a higher quality product. You get their expertise, you get their increased scalability, improved supply chain management. Perhaps your reaction needs a kilogram of potassium carbonate, and they have a supply chain to provide USP grade potassium carbonate for other companies that they do work for, and they have a way to buy it where it doesn't cost nearly as much, and they have a guaranteed supply. And you may have had trouble getting USP grade potassium carbonate, especially in the quantity you need. So supply chains do matter, especially when you go to bottling and injectables where you need vials, especially what we just saw in the pandemic, where BARDA had procured 50 and 100 million vials or 300 million, and nobody else could get any. We just saw this happen. So supply chain management does help. They have an established infrastructure, reduced labor, excuse me, better allocation of talent, dedicated project management, end-to-end options, and a partnership. They also frequently provide the manufacturing dossier for the FDA, usually at a fee, but you don't have to write it, and it's very detailed. For those of you who know the ECTD format and structure, it's Module 3. And they will frequently provide you a drafted Module 3 ready to go to FDA. It may charge you $300,000 for it, but it's worth it because it's written to FDA standards. So what are the top 10 manufacturing or contract manufacturing mistakes? Poor handling of unanticipated project delays and manufacturing capacity constraints. You didn't put rights to audit in the contract. You lost flexibility and responsiveness. Not basing some percent of the payment on quality of material or giving you the right to refuse it based on specifications not being met or regulatory inspection results. Scale-up issues, giving away too much product control during the contract negotiation, forgetting the benefits of a good contract manufacturing client relationship, being too lenient on setting the raw material quality standards, risking intellectual property loss, and not anticipating the differences, challenges in meeting scheduling, as well as culture of overseas companies. So there are entire companies, and I'm wrapping up. The next slide are the helpful links, which will show you links to the cGMPs for the phase 1s, format and content of an IND at phase 1, Q6B specifications, which are the test procedures and acceptance criteria for biotech and biologics, and M4QR1, which is the guidance on the format of a registration application for drug substances and drug products. So, having said that, the mistakes can be the risks can be mitigated. The mistakes are known, and you can work to minimize making mistakes. Entire virtual drug companies have been developed where the owner of the intellectual property never took possession of the drug. It went right from CMO through the process, through the testing, through the release stage, was shipped to the clinical trial sites and used in clinical trials, and the owner of the intellectual property, the drug company, was all on paper. They reviewed it on screens, they signed off, excuse me, and they never touched the material from the beginning until it went to the patient. They never touched it. And there are drug companies, and the FDA says it's okay, provided you take responsibility and you have appropriate controls. So I hope this has been helpful in a sense of what you can do virtually and with contractors so that you don't have to fear that you're going to have to start up making a GMP batch of material on your own. So thank you for attending, and I'll be happy to take any questions that we can answer today.
Thanks, Mark, for that presentation. At this point, if you have any questions, please submit those into the Q&A window. We do have about 10 or 12 minutes before the top of the hour. I do see one question that's come in, Mark. How does manufacturing decide on acceptance limit? For example, purity or microbiological limits?
As for microbiological limits, there will be guidance from FDA or ICH on what the limits are, what the testing requires. We go through those pertaining to the specific product and pull out what the limits are, and we'll tell you. It depends on if it's an oral dosing form or a sterile injectable, as well as purity is what are the impurities. Are you at 95% and that's the best you can give? Are you at 98%? Or did you have an issue that one of our clients recently had where they had to attempt to make a toxicology batch a little less pure so that they would see some toxicity? Now, that just screams jealousy when someone has to make a less pure batch just for toxicology purposes. So your purity has to do with what are your impurities and what type of product is it? And the rest of that answer is it depends. But you talk to FDA about it as well.
Thank you, Mark. I did want to draw everyone's attention to the feedback poll that is now opened. We'll give you a couple minutes to complete that, but we do greatly appreciate your feedback. Mark, I'm not seeing any additional questions at this time, but I did want to pause, give it another 30 seconds or a minute or so to see if anyone's typing their question in. Again, any questions for Mark, just submit those in the Q&A window. Thank you. And I do see another question has come in. This attendee says, thank you very much, Mark. I have a specific question about starting material. We would like to use a chemical available at a very high purity and in metric ton quantities from many vendors as API. How do we approach cGMP? Do we go back a few steps or reprocess the final material under GMP conditions?
I want to first say you can't apply quality retroactively. That said, it will depend, and I don't see the detail other than you would have to qualify it yourself. And while you would have to qualify it and you would have to assay it and go through checking for any potential issue, any potential impurity, and then you would talk to FDA. Now, if you are a raw material producer and that's the best material you can buy, you also check to see USP specifications and see if if the USP has written a spec on what constitutes, and you could compare yours to the USP grade material if there is one. So without knowing the full details, talk to us, but while you can't retroactively apply quality, you can check and make sure that the material does match the spec set by USP or EP if there is such a thing. Sometimes you just run multiple assays on the material. And I also saw a note about a final slide. Basically, beyond the helpful link slide is just a Q&A. So that was the end of the presentation.
Great, thank you, Mark. I'm not seeing any additional questions at this point. I did want to remind everyone that today's session was recorded and it will be posted on the Catalyze website along with the slides. So if there are no further questions, on behalf of the Catalyze team, we're going to close out the session and I hope everyone has a good day. Thanks, everyone. Thank you, Mark.
Thanks.